Hello, welcome to Chapter 14. In Chapter 14, we're going to be talking about someone named Mendel, Gregor Mendel, who did a lot of understanding how genetics work and understanding what genes are. In this chapter, we're actually going to talk about, this chapter and the last couple of chapters we're doing, we'll get to talk about a lot of actual scientists and kind of understand how science is working to understand genetics. A lot of this is, Mendel was a couple hundred years ago, but a lot of these scientists we're talking about are more recent even. Some of them are off, several of them are even alive still. And so this is a lot of very interesting, like these last few chapters are really interesting in that sector of these are things that you could actually go into science and still be studying. Mendelian genetics is probably a lot of what you think of when you think of genetics. If you haven't taken genetics classes before, making Punnett squares where you see, mom and dad's genes and what their kids' genes could be. So we're going to have some fun doing some of those. So the underlying question for this chapter is, what principles account for the transmission of traits from parents to offspring? One of the ideas is a blending hypothesis. Blending hypothesis says that your genes from your parents just blend together. So if you had blue paint and you have yellow paint, they're just blue paint and yellow paint, they would blend to make green. So if you had a parent with blue eyes and a parent with yellow eyes, they would just blend together and give you a child with green eyes. The particulate hypothesis is that each parent passes on discrete heritable units. So not that I take from mom and dad and just blend it into a new version for me, that I have copies from mom and dad that are going to be expressed in their own unique ways. And so this is a study that Mendel was doing. Mendel was a garden monk. Let's see, this is Mendel right here in this picture. He was a monk and he was doing experiments. At that point, almost all science came from clergy. And so he was doing these experiments with garden peas. If you've ever grown peas in your own garden, they make flowers that look like this. And he was looking at what color do the flowers turn out? Are they tall or small? What color are the little seeds, the little peas themselves? What shape are the pea pods in? And so he did all kinds of studies to examine how genes are passed on from parent to child. So he did these carefully planned experiments where he breeded these different garden peas to discover these principles of heredity. He kind of understood the basic principles, and from here we're going to go into some different, exceptions and kind of a little bit more nuance to some of these hereditary rules. So Mendel's approach let him deduce principles that had been elusive to others. And so to back up a little bit, let's talk about some different terms. So character is one term you need to know. A character is a heritable feature that varies among individuals. So he's going to look at the character of flower color. So this is a heritable feature. Purple flowers typically would give rise to purple flowers. White flowers would give rise to white flowers. It wasn't just randomly. Where is it called? Okay, and then a variant of each character is a trait. So purple is a trait, white is a trait. So character is flowers have colors, the trait is purple color or white color. Peas were available to Mendel in many different varieties. They grew quickly, they grew reliably, they were easy to propagate. So he used pea plants. And so this is what he saw. He had a purple flower and a white flower that he took and fertilized together. So he used this little paintbrush to paint pollen from one plant to the other plant. And he found that these plants, he collected the peas, which are the seeds, and planted them, and he found all of their children all had purple flowers. So a white flower and a purple flower blended together to make purple flowers. So if we back up, does that sound like a blending hypothesis or a particulate hypothesis? So this doesn't look like you've gotten like a halfway between purple and white. It's not kind of a light purple, so that kind of says it's not blending. This suggests that there's something heritable. They're getting white genes and purple genes and expressing them as purple. So white and purple gave rise to purple plants. Mendel chose to track characters that only had two distinct alternative forms. So the flowers didn't exist in a range of colors. They were white or they were purple. The seeds were yellow or they were green. They were tall or they were short. So they were two very distinct forms. It couldn't be, well, it's kind of this, kind of that. It's very distinctly this or that. And he also started with varieties that were true breeding. So true breeding meant that purple always gave rise to purple flowers and white always gave rise to white flowers. He didn't have any hybrids. He had like genetically pure, I don't really like that phrase, but pure plants that, all the purple flowers only had purple genes, the white flowers only had white genes. And he knew that because after breeding them for several generations, the purple flowers only would give purple and the white flowers would only give white flowers. So they were true breeding. Their plants would always have offspring of the same variety when they self-pollinated. So in most of his experiment, he would mate two different true breeding varieties. So a purple flower and a white flower, a true breeding purple, a true breeding white flowers. And that would give him a hybrid. So a purple and a purple true breeding, a white true breeding, hybridized together to make a hybrid. So some generation names here. P generation is the parents. The P generation is always true breeding. The hybrid generation is the F1 generation. And then if you take F1, that means first filial, so they're the brothers for each other. If you take those F1 and cross-pollinate or self-pollinate F1 individuals with other F1 individuals, you have the F2 generation, so the second filial. So it's like the parents, the kids, and the grandkids, okay? So when he crossbred two distinct true breeding different traits, so a purple flower and a white flower, all of the first generation were purple, no matter what. They were always purple. But then if he took all of these hybrid purples, and had a second generation, he would see both traits start showing up. But they showed up in a certain ratio. You would see 3 purple plants for every white plant. And this is going to be kind of important, this 3-to-1 ratio. We're going to come back and talk about why that ratio shows up. But right now, Mendel didn't know why that was showing up. So we're just going to hold it at true breeding, 2 different characters. 1 character would be what showed up. And then in the second generation, 3 of them would look the same as a hybrid, and 1 of them would look the same as the other parent. So Mendel reasoned that purple flower factor was affecting flower color in F1 hybrids. So for some reason here, the purple flower is what is determining flower color, and the white flower color is not able to determine white flower color. So he called purple dominant and white recessive. And to this day, we discuss dominant and recessive traits. So because if you took 2 true breeding characters, whatever shows up in that first hybrid generation is the dominant trait. So because the F1 generation were all purple, purple was the dominant trait. But the white color was not getting destroyed here. So the white color is not getting destroyed before you have your, during the F1, because it's still able to be passed on and show up. So this purple plant is still holding on to that white color, that white gene, we'd say today, but it's not able to express it. That purple gene is overpowering it and dominating to control the color. But it's not diluted and it's not destroyed because that white color can reappear in the F2 generation. So he looked at these 6 other characters. So he looked at purple and white flowers, and he found 3 purple to 1 white. He looked at yellow seeds versus green seeds, and he found a 3-to-1 ratio. He looked at seed shape. Are the seeds round or wrinkled? In the F2 generation, he also saw 3 to 1. So round is dominant to wrinkled, yellow is dominant to green. What color are the pods? What shape is the pods? Are they inflated or are they constricted, kind of wrinkled? Where are the flowers located? Are they axial, so in the middle of the plant, or terminal, only at the ends? Are the stems tall or dwarf? Are they tall or short? And for all of these, in the first F1 generation, he would see only the dominant trait. And in the F2 generation, he would see 3 dominant for every 1 recessive. So Mendel discussed a heritable factor being passed from the P generation to the F1 and from the F1 to the F2, these heritable factors. And today we call these heritable factors genes. So he developed a hypothesis to explain why he sees this 3 to 1 inheritance pattern in the F2 offspring. And there are going to be 4 related concepts that make up this model. And these concepts are still applicable to what we know about genes and chromosomes today. So we are going to say this is your gene. You have 2 copies of this. You have 2 copies of each chromosome. So plants have 2 copies from their dad and from their mom. From one parent they got a purple flower gene. From one parent they got a white flower gene. So these are their 2 different alleles at these particular locus, at this locus on each chromosome. So here's the DNA for purple flowers. That makes a purple flower gene. Makes this purple pigment. The white flower allele does not make a pigment. So that ends up with a plant that makes a purple pigment. And that purple pigment turns the flower purple. Then as this cell goes into meiosis, they might end up making a cell that has a gene for the pigment or doesn't have a gene for the pigment. And the next generation to decide if that next generation turns purple or white. So these 4 concepts about what Mendel elucidated. One is our alternative versions of genes. And those different versions of genes give different versions of inherited characters. So there is purple versions of genes and white versions of genes. And the gene for flower color in pea plants exists in those 2 versions, purple flowers or white flowers. Those 2 different versions of a gene are called alleles. So you can have the allele for purple flowers or the allele for white flowers. This is a term that is going to show up for the rest of the semester, so get comfortable with that term, okay? And each gene resides at a specific locus on a specific chromosome. So whether you are the purple gene or the white gene, it is always going to be found at this spot of that chromosome. No matter what flower you are, if you pick out that chromosome, you have a gene color allele at that location. Second, for each character, an organism inherits 2 alleles, 1 from each parent. Mendel made this deduction without knowing about chromosomes, but you get an allele from your mother and an allele from your father. True for animals, true for plants, true for all sexually replicating organisms. The 2 alleles could be identical. That's what we see in true breeding plants. In a true breeding plant, they have 2 purple genes, so they always pass on purple genes, and the next generation has 2 purple genes. In the white true breeding plants, you have white genes being passed on to make white flowers. Sometimes you don't have a true breeding plant. You have 2 different alleles. That's what we saw in the F1 generation with the hybrids. They had 1 copy of the purple from their purple parent and 1 copy of the white allele from their white parent. Third, if 2 alleles at a certain locus differ, then one is going to be dominant, and the dominant determines the appearance. The other allele is going to be recessive and doesn't affect the appearance. So in those flower colors, the F1 plants had a purple allele and a white allele. The purple allele was dominant. It determined that those flowers would be purple. The white allele was not dominant. It was recessive. The white allele did not affect the plant's color. Fourth is the law of segregation. So if the 2 alleles for heritable characters separate, which is what happens during gamete formation, they end up in different gametes. So an egg or a sperm only gets 1 of the 2 alleles that are there in the organism. So I have the allele, this allele and that allele for type A blood and type O blood, but only one of those genes is going to be passed on to my eggs. Those flowers the hybrid F1 generation had an allele for purple and an allele for white, but only one of those alleles got passed on to their next generation. And the segregation of alleles will correspond to distributing those homologous chromosomes to different gametes in meiosis. Worth noting at this point that mechanisms of mitosis and meiosis were not yet worked out when Mendel was doing his garden work. So Mendel kind of laid the formation, the groundwork for us to really start understanding what is going on, what is actually happening during meiosis. So this model accounts for the 3-to-1 ratio in the F2 generation. So purple flowers only pass on purple alleles, white flowers only pass on white alleles. That means our F1 generation are going to have both the capital P, the purple, and the lowercase p, the white alleles. In their gametes, half of their gametes are going to carry on the capital P, which is the purple, and half of their gametes will carry on the lowercase p. And then if we look at the F2 generation, so the F2 generation, their sperm could be either purple or white alleles, and the eggs could have either purple or white alleles. That means there's 4 different versions that we could have here. One version is you get a purple allele from dad and a purple allele from mom. Those flowers are going to be purple. One version is you get a purple allele from mom and a white allele from dad. That means those flowers are going to be purple. One is you get a purple allele from dad and a white allele from mom. Those flowers are still going to be purple. And one is that you have a white allele from dad and a white allele from mom. Those flowers are going to be white. So this is a point in class where we would stop and actually work through some different Punnett squares. And I'm going to have to leave this to you. If you're not attending class today, you're going to need to come up with some Punnett squares. You can just look on the internet to kind of understand how a Punnett square works. This is kind of important. So this is why we see this 3-to-1. So if they have 2 copies of the dominant allele, or if they have 1 copy of the dominant allele and 1 copy of the recessive allele, these are 3 different ways to have a purple flower to 1 way to have the recessive trait, the white flower. When we are discussing dominant and recessive, we always write the dominant in a capital letter and the recessive in the lowercase letter. So here it's the letter P for purple. So capital P means purple, lowercase p is going to mean a white allele. Okay, an organism with 2 identical alleles for a character is called a homozygote, and it is said to be homozygous for that gene. So you can be homozygous for one gene and not homozygous for another gene, okay? But when we are talking about one particular gene, you are homozygote if those 2 alleles for that gene are the same. If you have 2 different alleles for that gene, you are a heterozygote. So it'd be heterozygous for that gene, for that character. So homozygotes are always going to be true breeding and heterozygotes will not. So if we back up a slide, go back to here. If you have 2 copies of the purple allele, you are homozygous for flower color. If you have 2 copies of the white allele, you are homozygous for flower color. If you have 1 purple allele and 1 white allele, you are heterozygous for flower color. And here you're heterozygous for flower color. So these homozygous purple flowers are true breeding. These homozygous white flowers are not true breeding. I mean, the homozygous white flowers are true breeding. These heterozygous flowers that have 1 purple gene and 1 white gene are not true breeding. Those are hybrids that can go on to have either purple or white characters passed on to their next generation. Okay, an organism's traits do not always reveal its genetic composition. So when you look at a purple flower, you don't automatically know if it's homozygous or heterozygous. It could be homozygous with 2 copies of the purple allele. It could be heterozygous with 1 copy of the purple allele and 1 copy of the white allele. So we are going to distinguish between phenotype and genotype. Phenotype means what it looks like, what it can do. So the phenotype for all of these flowers is purple. Phenotype for this flower is white. The genetic makeup is its genotype. So homozygous or heterozygous is part of your genotype. You have two dominant alleles, that is your genotype. You have a dominant and a recessive allele, that is your genotype. You have two recessive alleles, that is your genotype. Phenotype, what you look like, what you can do. Genotype, what your genes are. So phenotype is physical trait, genotype is your genes. Okay. An individual with a dominant phenotype could be homozygous dominant or heterozygous, and you might not know. So to find out, you can do a test cross. In a test cross, you take the person with an unknown genotype and cross it with, well, you wouldn't do this with a person, that's a bad example. You could take a dog or a flower, an organism with an unknown phenotype, and breed it with an organism that is known to be recessive. It's known to be homozygous recessive. And then you look at their children. So if the one unknown parent has a genotype of two dominant alleles, then all of their children are going to be dominant. If they have a dominant and a recessive allele, then they're going to be half of their children dominant and half of their children recessive. So I want you to pause at this point and see if you can recreate this. So the eggs can either be all capital P, all dominant, or the eggs could be half dominant and half recessive. You don't know at this point which type of genes this organism has. So when you cross it with the recessive ones, if this is a homozygous dominant, then it's passing on a capital P to all of their children. So all of their children are going to have a capital P gene. They'll all be purple. If this unknown organism is heterozygous, then it'll pass on the dominant gene to half of their children, and it'll pass on the recessive gene to half of their children. The recessive organism only gives a recessive gene. So then you end up with either half of the cells having dominant from mom and recessive from the recessive phenotype, or being both recessive, getting a recessive from mom and a recessive from dad. So then you can stop and look at what the individuals look like. So if you have all purple flowers as a result of this mating, then you know that this individual is dominant, homozygous dominant. And if you get half of them purple and half of them white, that fits this Punnett square, so you know that this individual is heterozygous. Okay. We are going to pause at this point, and I will come back to chapter 14, part 2 with you later.